Good evening,
All right.
everybody. So today, Vlad Vinogradov will make a very interesting talk. He will present the gold medal top 8 solution for the competition to predict the binding of the small molecules to the proteins, which recently was on Kaggle. There are more than 1,000 participants, and Vlad was at the gold zone. So thank you very much, Vlad, that you agreed to make a talk, and please start.
Thank you, Alexander, for the introduction. So I'm Vlad. I work at Bioptic. My role is CTO, but you can imagine I'm doing a lot of technical things as well as the business things. So I talk to clients all the time. And meanwhile, I find time to actually try to develop some new neural networks and overall apply deep learning for the field of drug discovery. For as the recent result, I got a gold medal on this competition. I would say that the main topic, the main idea of my talk on the solution will be about the benchmarking. So let me first introduce myself a little bit more. So I am a computer scientist by training, and I have been working at ITCS in the past, which is a spin-off of Intel, and then it was acquired by Intel again. And the next year, I also was working on Intel as an intern. And a few years later, when I was getting my data science master degree, I worked at Bayersdorf in Hamburg, Germany, where we were applying deep learning methods for recognition of facial biomarkers. Then I turned to be a co-founder and head of AI at Yora.ai. It's a company that was creating many B2B solutions based on machine learning. So it was kind of a research, contracting, service company, all of a mix. And then we finally pivoted to Optic, and Optic is also a startup, and we started it with the basis that we are able to build high quality image retrieval systems, and that's not related to drug discovery at all, as you can see. But we started with this, and we found a client called OpenSea that was very that loved our technology for building which we built for image deduplication. Basically, we were comparing whether a similar art is in the space on the blockchains, and that's very different from what we do now. But this is what we started with, and when we got our investments, we got them exactly for this idea. But later on, we discovered that we are not so motivated to actually doing trust and safety in general, because that was actually the only path for us to go. And we decided to pivot to something big, and we saw that the machine learning for drug discovery has not yet arrived. And I mean, in terms of the foundational models that we see now in natural language processing and computer vision, right? Audio as well. So we decided to switch to this domain, and that happened a year ago from now. And I was already able to get 2 gold medals on Kaggle in this field. One is the previous one was about the protein function prediction. Anyway, this one is about small molecules. So let's switch to the topic. For those who did not check the overview of the competition and maybe doesn't know how the data looks like, I will give you a short video introduction that I liked, was shared by some of the participants in the competition. And this video will give you an introduction of how the data is collected because this competition is about predicting binding affinity in a binary sense, whether a molecule binds to one of 3 targets. So let's see how Leash Bio collects the data.
Screening novel compounds that interact with biological targets is of key interest to pharmaceutical and biotechnology companies and research institutes. Traditional high-throughput screening, HTS, is limited to millions of compounds. Screening billions of compounds is entirely beyond the conventional HTS approach. HitGen's DNA-encoded library technology can screen over billions to trillions of compounds efficiently. To build a DNA-encoded library, we begin with a scaffold as the basis for each compound. To this, a chemical building block is added sequentially, followed by a corresponding DNA tag. Further building blocks, along with corresponding DNA tags, are sequentially added to create the desired compound design. The complete DNA tag acts as a unique barcode for each compound, recording the building blocks and chemistry used. Instead of building all compounds individually, HitGen uses a highly efficient split and pool process. At each synthesis step, compound intermediaries can be split, so different building blocks can be added. However, intermediaries that share the next building block in common are pooled for reaction efficiency. This split and pool process can be repeated for different building blocks, so that vast compound libraries can be made efficiently and economically. Because each compound has a unique DNA barcode, all compounds can be pooled into one library, which is now ready for screening against a biological target. The protein target is then added to the DNA-encoded library of compounds. After allowing time for interaction, compounds which fit the target well, because of ideal hydrophilicity, lipophilicity, and molecular geometry, will stay on the target. Compounds which don't fit well bounce off. After the washing step, compounds with strong affinity remain and are then released off the target and collected. Using the DNA barcode, the compounds are identified via amplification, sequencing, and decoding. The identified compounds are resynthesized and validated to establish whether the protein target activity is affected. Once chemicals have been validated, their chemical structures can be optimized to generate novel clinical candidates with improved activities and properties. HitGen's chemical libraries are synthesized and screened efficiently, and the available compounds for screening is expanding from the billions into the trillions. HitGen's DNA-encoded library platform has focused on creating more chemical diversity, improving compound drug-like properties, and enabling more screening options. This vast resource is highly valuable for innovative drug discovery and meets customers' needs great and small.
As you can see, this is a different company who created this video, but a process is similar, at least in the part of the building blocks and how they fit into proteins when the screening happens. I will quickly go through the overview of the competition so that we also have everything in place and like see at least what data we have. So Leash Bio collected in total 133 million small molecules, and they build of building blocks, and they screen them against 3 targets. So there is a nice picture how the data looks like. Let me find it. Yes, here it is. Basically, each molecule has 3 building blocks, and typically they are connected through a core. And the interesting part is in this competition is that for the training set, there was only one core given, which is triazine core, basically 3 nitrogens in a circle with carbons, and all other building blocks are connected randomly to this like connecting points, basically carbons within this triazine core. And for the test set, they gave around 38 new cores, and it's kind of something that already shows us that, well, the data set that will be used for the testing is quite different from what we have for the training. So that's the first assumption that we need to that's the first like idea that we need to take. Maybe we should not like build a model based just on building blocks. And the process is similar here, and they tested it against 3 targets, as I said, which are, let's go through, which are SEH, BRD4, and the HSA. Basically, well, one of them was for cancer, the other one is for the blood pressure and diabetes progression, and one of them also, if I remember, let me check quickly. And another one is typically anti-target, and it means that we should not bind this target later on when we optimize our compound. So that will be used, but that might be used by our models might be used by organizers to also check the compound for not to bind a certain target. But anyway, the model will be the same for all the targets because we want to model this activity prediction. So now I hope this shallow introduction is enough, and I hope if you want some more details, you can go to this page and then find it. And now let's switch to the solution itself. So first of all, I want to say how the evaluation is performed on this competition, because it's so important to know that. And for that, let me quickly check the discussion that organizers put. That happened actually in the middle of the competition, and before that, there were some like unclear moments on how the evaluation is performed. And then after discussing with a lot of participants, organizers published a scoring method, and they finally showed the new leaderboard and how it looks like. And it was now clear that how we should progress in this competition. First of all, there are differences for public and private metrics, and for each target, the picture is the same. But for the public metric, there are 2 kinds of splits: shared building blocks and non-shared building blocks. What that means is that, in fact, we have in our test set blocks that are present within the training set. Like a lot of the blocks are intersected between training and test set, so that will be a shared part, and typically it is sampled randomly or by scaffolds. And for the non-shared part, that's the tricky one. So basically, organizers select a few building blocks that are not present in the not given in the training set at all, and they ask us to generalize for the building blocks that are not in the training set. So you can and the global macro average precision is calculated. So you can quickly find that 50% of the metric will be dedicated to shared building blocks and 50% to non-shared. On the private leaderboard, although and there is a new library also added, which adds which takes another portion of our metric, right? So this time it's 33% is dedicated to shared building blocks, 33% to non-shared, and 33% to the new library. A new library is that part of the chemical space where the core is different from what we have in the training set. Those are 38 cores that I was talking about. So this is this also highlights that, well, we don't need probably to train the model just on building blocks because the core might affect the results so much. So we better switch to training a model globally on a molecule, and whatever we take as a model, but we need to train a model that is, that captures the whole molecule. That's at least how I see it, and how I was performing during the competition all the time. So let's switch to my solution. First of all, I can tell you that this is a mostly like a single. I call it single because the architectures are quite similar. So this I basically came to a conclusion that smiles-based transformer model can perform very well on this task compared to other models, and it actually outperformed my other experiments. And the final solution only has two kinds of smiles-based transformer models, which are Roberta and Molformer. Roberta is a typical NLP model that was developed quite a few years ago, and people now started applying it to smile strings with Camberta, maybe one of the known models in this space. And later on, Molformer was developed by IBM, and actually they published the checkpoints of the pre-trained model as well. They have trained it on 1 billion smile strings, but they only published checkpoints for 100 million molecules. So it's kind of not the full checkpoint that we would get, but still it can be used for the fine tuning, and that actually could improve the quality. So overall, the solution is like, first of all, as we have shared building blocks part and non-shared building blocks part, we can predict separately for shared and non-shared. It probably doesn't make sense to predict on the new library also separately. I mean, it might be, but it is kind of hard to model because we don't know any labels, how those cores, 38 cores, affect the binding affinity. So it's maybe easier and also maybe just better to focus on what we have in the training set and work on those molecules. So let's have shared and non-shared predictions differently, so that for the test set we cut our prediction. We cut the test set into halves, shared part and non-shared part. That's it. And as you can see in other solutions, some other teams also did the same. And now our task is to develop the best model for each of these halves of the data splits. And the obvious thing comes into mind that for the shared building blocks, you might just need to overfit. And we need to train it on all data, most likely. But for the non-shared, we better need to generalize to the novel subspace of building blocks. So let's somehow find a way to do that. That's the hardest part, I would say, and everybody was struggling with this. So that's why I spend a lot of time on data splitting in my solution. First of all, it's a very known problem that may be not so clear to everybody in academia, maybe to industry as well. But people started realizing that the current data sets that are publicly available for small molecules property predictions are actually biased by similarity. Let's say they typically split the data randomly, and what they get is that in the test set there will be a molecule that is just one atom change from the molecule, from the closest molecule in the training set, and that is that actually is a huge bias, and the models can quickly realize what actually parts of the model or molecule affect their binding affinity and other properties, and as there are most likely will be the same properties for the molecule with one atom change, the models just overfit and easily predict those. So that's why community started doing some efforts in the direction of creating better benchmarks, and Polaris actually has been announced a few weeks ago, and that's an initiative driven by Valence Labs, and it has a huge community of biopharma companies. And the idea here is that, well, let's have something like Hugging Face, but for the drug discovery field with two things mostly: data sets and benchmarks. So data sets are everything that we can publish publicly or privately, and benchmarks is data splits mostly that are set for these data sets. So it's an open source tool, and there are a few data sets already available here with different, within different domains. Basically 83 data sets in total, and let's say we select ADMET, there are like a third of them is for ADME prediction properties, and yeah, also they put benchmarks and even more of them. So again, there are different data sets and benchmarks here, and everybody is welcome to publish their own benchmark. So that was an introduction to the problem itself. And now let's switch to what we have in the papers. And also I have to put this ad, but this is what I'm grateful for my colleague who actually published the paper recently called Low High. It's a benchmark solution where we can prepare the complex splits of the data set for molecules so that our training set and test sets are very different from each other. Basically, they are dissimilar by Tanimoto similarity. And he calls this split hit identification split, because typically when we work with the real screening campaigns, we try to find a novel compound which is not similar to what's already available in public, and that gives us that actually restricts us to use some threshold on the similarity, and FDA agents who accept the patents, they typically have in their mind something like 0.4 Tanimoto similarity cutoff between the two molecules when they try to assess they are similar or not. And for this purpose, to perform such a split, we have to work with similarities between each molecule within the data set, and it's solved with a graph modeling. Let's quickly go through this method to have some idea. So if we consider a graph G of vertex of set of vertices V and edges E, where each vertex is molecules, just its molecule in the set, and V is a and the edge E is set between these molecules only if they have similarity above certain threshold. So let's say above 0.4 nanometer similarity. We consider those molecules as similar. So our patent agent will not accept, this proposal of a new molecule if it is similar by more than 0.4 nanometer similarity. So two vertices will have an edge if the similarity is above 0.4. And the similarity is calculated by tiny motor similarity with ECFP for fingerprints. But the problem is, in this graph, we will get a huge component of 95%, where typically 95% of molecules will be dedicated to one huge component, and we cannot break it up. And to do that, we can apply a known algorithm called balanced vertex minimum k-cut solution. It's a linear programming optimization where we can find a few like as less as possible dots, like molecules within our graph, that if we cut, like remove completely from the graph, they will relax our structure in such a way that we will get the normal components of the graph, let's say to have 80 to 20% split. Basically what people you typically like, right? Let's have 80% for the training set and 20% for the test set. So we can get those size constraints when we relax the problem, when we remove a few molecules from the graph itself. So yeah, kind of we'll lose some points, but typically it's a very small fraction of them. So if you apply this algorithm, by the way, it's available on GitHub. It's called low-high splitter. So if you apply it and on your data set, you might get just a few data points removed. That's not bad. You can start working with this highly difficult data splits. So are there any questions at this point? Don't hesitate to ask at all. Like comfortably can discuss it.
Colleagues, any questions? So don't hesitate. So as far as I understand, the idea is to split by similarity, yes, the molecules such that kind of similar will be in one fold and in the test fold will be very dissimilar, yes?
Yes.
Okay.
Yes, this is the idea. Yes.
Yeah.
Quickly. So yeah, and that actually models our problem much better now in terms of generalizing to novel subspace, what organizers ask us to do. So yeah, that's the idea. Well, if you have any questions along the way, please raise your hand, whatever we can discuss.
Just as a remark, we had before the talk by the author of this paper, and the video can be found on the YouTube if someone wants. Interested.
Oh, nice.
Okay.
Oh, nice. Yeah. So Simon presented this work. Cool. So now let's.
Hello.
Yes, please.
Hello, Vlad. May I ask you? As I see, you used ECFP features, right? Fingerprints to calculate the motor similarity, and why not others?
Oh, good question. So some people use max features for calculating the similarity, and I heard that they are good for modeling similarity as well. I mean, they are good for modeling similarity, but they are bad for building models. Let's say if you want to train an XGBoost model out of the features, typically ECFP win. But indeed, for the similarity, we could use different other methods. And I agree that we could try, but I did not try it. And also that's because I didn't want to change the algorithm itself as it was built by Simon and which is published. So yeah, that might be an interesting idea to explore.
Okay, thank you.
Yeah, thank you for the question. Bring more.
Yes. I also have a question,
Right.
maybe a silly one, but I didn't catch an idea how building new graph model where V represents molecule and H represents the similarity between test and set between most similar molecules. It should be before threshold. And how collecting this graph helped to build a new benchmark test. So
yeah. Oh, yeah. All right. And now I will switch to how I build the benchmark itself. Yeah,
okay.
that's what I wanted to go for. Yeah. And thank you for pushing me. So let's continue. Indeed, how can we apply this algorithm to our data? First of all, let's look at the similarities between not the full molecules, but between building blocks. We have about 2,000 unique building blocks in total, and which turn to 100 million, molecular library because of the like possible combinations. And I specifically started looking at the building block similarities because even if two molecules might be very different, but there might be just one change in a building block. And that's what I want to get rid of. I want to model the similarities on building blocks level to be more precise, because we are tested on the building block splits, not the molecule splits, right? So let's look at the distributions of similarities between training and test building blocks, so that we have a sense like how our training set is different from the test set. And we see that, let's say we compare building block at position 1 with building block at position 1 between the training and test sets. And we quickly see that, first of all, a lot of duplicates are present. So tiny motor is just 1 because this is what the shared part is about. So we do have a lot of similar, like not similar, but the same building blocks between the training and test set. This is for the shared, so we will consider them for the shared building blocks part. We will not work with them in the scenario of this hit identification data splits, because that's not our main goal in this subset of data. For the shared part, it's a different story. So for the non-shared, we will focus on the similarities below 1. And we will try to split those building blocks by similarity. So on the diagonal, let's continue. We have BB2 compared as well, and we have a lot of duplicates, BB3 as well. Let's see how the cross building block similarities look like. So building block at position 1 to building block at position 2, where BB1 is from the training set, I believe, and BB2 is from the test set. Or maybe vice versa, but doesn't make so difference because we have both. And we can see that the most of the similarities are below 0.4 tiny motor similarity, so they are very, very different. So that's fine. BB1, BB3 are also very different from each other. What we can say about BB2, BB3, because they are so similar, and many duplicates are between the two sets as well. So those we will actually merge later, so that it is easier to treat the problem, and to not have any leaks between different positions. And now let's focus on the part where we have similarities below below 1. The question here is like how to split. Basically, I described the method, and now let's put the distributions between in the training set itself. So because that was for the between the training and test set, and now we want to see what we have in the training set, so that we will create our cross-validation data splits, or maybe just data splits. So for building block 1, the mean of similarities is around 0.7. So for the building block 2 and building block 3, the mean similarity is about 0.4. So they are very different. I mean, the distributions are different between BB1 and BB2, BB3. And that's why we need to adapt our threshold per building block position. And here, a red vertical line shows I actually put the threshold that I chose for each of the building block positions. And I selected them to be close to the mean, although I don't have any like science around that. So it's kind of something that I wanted to try, and something that actually works in the linear programming solution. Because if you move the threshold too far away, that might even not have any solution, and the minimum k-cut problem will not be solved. And if you move too close to 1, we may get too easy data splits, again with a lot of leaks. So I just took a mean. Some people may explore different strategies. And yeah, as I said, we merged BB2, BB3 because they have similar distributions, and they also, have some duplicate leaks between the two sets. And we only apply high splits on sorry, here, it should be BB1 here. So we only apply on BB1 splits and BB2, BB3 as a joint set. And when we try to split, let's imagine how that will look like. If we plot if we plot a cube with 3 axes where a few building blocks, 3 building blocks are presented, we will see that we kind of dedicate certain building blocks in combinations to the training set, and certain building blocks in combination to validation set, and everything else has to be removed. So that's not like the drawback of the algorithm itself that I presented before. That's the second decision that I made. Basically, I wanted to not have any similar building blocks in all other positions. If I chose some building block at let's say at a given position. So basically, the idea is we need to have a so spread building block sets in conjunction. So building blocks, like basically all 3 building blocks. Well, I hope you understand this picture clearly, but the idea is let's just have them so diverse and not try to. If I chose building block 1, let's say this one, I chose it for the training set. I should not have in validation set anything that is on the lines by all 3 dimensions to this building block 1. That's why I am removing everything in this cube. So you can see my validation doesn't intersect with the training at all. So that's the hardest data split that we can apply here. And of course, it removes a lot of data, but likely we have a lot of data in our data set. It's as I said, it's 100 million libraries, so that's plenty of data. And by the way, there is about 1.5% of positives, and the rest is negatives. But that's also like enough. And this data split is very hard. When you try to train a model, you will quickly see that nothing works. That's what I wanted to show you here as well. I collected a few other plots that are not in the solution. So let's see it on the example. First of all, let's have a random data split and train two models on the random data split and validate it as well. And we will see that the model that has more parameters will typically overfeed, right, over the model that has less parameters. But here we will see that, well, indeed, like the model with much more parameters tend to have higher score in the training set earlier than the model with less parameters. But the most important part is that they match the quality on the validation set, close to the end of the training. And on the training set, of course, the model with more former with more parameters overfeed. And when we apply the hit identification splits, called high BB splits, we will see that models actually don't progress on the validation set. I put different models here because I didn't find experiments in the same setup with Molformer and Roberta. So please just think of them as I will show you the difference later, for those who have questions here. But anyway, the thing is that
step x-axis. Yes.
When it's actually batch step. So we put a batch. Yeah, it's a training step.
Yeah. And so you are fine-tuning these models. Yes, is it correct, or you are training them from scratch, or?
Oh yeah, I fine-tune Molformer because I have a pre-train, and I don't fine-tune MPNN++ because I didn't have checkpoints publicly available for this architecture.
And then what is the step for more MPNN?
It's the same, like basically, but they are in different. They have different batch sizes. That's why there is a different scale. Basically, the same amount of epochs, let's say, but different batch size.
Mm-hmm. But so you say that the conclusion is they do not kind of perform better with the number of steps. Yes, is it so? They are not improving. So
they first of all, yes, they are not improving, and they are fluctuating a lot, as you can see on the validation.
Okay, so that means we can take them as it is without any fine-tuning. Yes, or.
No, the point here is that compared to the random split, we got around 60% average precision for on the random data split. But when we turn to the high splits, we quickly get much worse quality. It's just, it doesn't go above 1% of average precision. That's what I wanted to show.
Okay. So what does it mean? It just doesn't work at all?
Yes, it basically means that the models don't progress. They don't generalize well. They don't work basically in the scenario of the novel subspace, on the novel chemical subspace. So, and that's important thing. That's actually what you will find for any kind of model. Of course, some of them will be better than the others, but in general, you will most likely see these fluctuations. And yeah, that's why Leash Bio organized such a competition, because they want generalizable models. Nobody built generalizable models yet. The whole point about like building them, maybe we need some foundational model retrained on like trillions of smile strings or whatever.
Okay.
Yeah.
And
So and you can see
if it's kind of almost do not predict, so what do you do? So it's don't use
so
it or
first of all, we can already try to find a model that will somehow stabilize during training on the validation set. But what I found is maybe this data split is so hard for this data set, and I decided to create one more data split. But from here, I selected the model that at least more stable. So I basically picked more former from these results. So this is one of the models in an ensemble that I used.
Okay, I see.
Yeah, and as I did not have well, and yeah, a good comparison here as well. Basically, you would say, oh well, maybe MPNN performs better here because it has some peaks with higher quality. Let's pick one, but turns out that that is so unpredictable. And when I pushed these models to the leaderboard, I actually got what I expected. But on the public set, it wasn't actually clear, but it was clear to me, like because of these fluctuations. And I decided the model that didn't perform better on the public set, yet it wasn't performing better on the high BB splits as well, but it was more stable. So, and I picked the mole former here, and it shows that on the basically this is for one target. So this is HSA target. I needed to put that, and that's why the score is not so like high. But anyway, this is when I just submit my predictions for one target and only for non-shared building blocks. So, and we see that Molformer performs 3 times like better than MPNN++, right?
By the way, don't you have such kind of comparison for other models? I don't know, from other participants. So here is Antonino, for example. Antonino, did you compare something like that, or Vlad, maybe?
Yeah, that's like I compared a few other models myself,
Okay.
and I have them in the solution. I wanted to also show you that. Let me check. So basically, Antonina asked the question like how these splits actually improve the quality, and I also like performed a few other scorings and got like, let's say, three models. They are different, but they might be compared to each other. So first of all, let's say we take a model Molformer trained on all data, basically 99% of all almost 100 million data set randomly splitted, and we submit this model for the shared building blocks part, and also test it on the non-shared, just in case to have some metrics. And we see that for this one that was just overfit on all data performs the best on the shared building blocks part, as expected. So this is a model that we try to overfit to our training set because the same building block will be in the test set, and we incorporated the whole data set to do that. And yet this is the worst model on the non-shared building blocks. And when we go further, let's say we take a model from the high BB splits. Here it is. We we checkpoint early. We checkpoint at the moment where we have the best score on the high BB split, so that we don't overfit, but the model is the same. Also, we actually reduced a lot of, like, this is done on the part of data that is balanced. Basically, I originally took a balanced positive negative data set, and out of it I created the training set. So that's much smaller than this one, because this one has a lot of negatives because of the natural distribution. And this model performs the best on the non-shared part, as we wanted to achieve, and it performs the worst on the shared part. So that kind of highlights that indeed these data splits are important for building models on non-shared building blocks. And also in the middle, I have another data split, which is BB split, which I wanted to discuss later. It is a simpler version of hyper split, and it performs, somewhere in the middle. Yeah, questions here?
Just to summarize, to check that I understand correctly. So, do I understand correctly? So you choose the model best model according to, for example, high BB split, and then you what you present here are the results on the private, public. I mean, all this combination. This is yes. And your conclusion is that if you choose by high BB split, then you see the best results on the private, non-shared.
Yes.
Both. Or yes.
No, just non-shared. Just non-shared. Yes, indeed.
Just non-shared. Okay.
Because and that's why I created this two-half solution, where I put one type of models for the shared building blocks and one type of model for the non-shared. That's the idea. Here we try to overfit for the non-shared training on all the data, and then for the non-shared building blocks, we try to apply different data splits, and one of them is high BB.
Okay. I see. Thank you.
Yeah, and the last piece and the last data split that I also applied is building block split. It's similar to what other participants did and what organizers did, as we all I kind of understood it, and but it is different in a few aspects in my case. First of all, I'm creating the 5-fold splits. So this is a proper cross-validation data split. And again, we create the exclusive. We somehow dedicate building blocks in the exclusive manner as we did before. So if some building blocks appear to be, let's say, in the validation set, those building blocks in any position cannot be in the training set. So we basically remove everything else. That's why we have this red colored subcubes, intersecting, like basically planes intersecting at the combinations of the building blocks in the validation set. So, and we do that for the 5 folds with different validation set taken every time. And here you can see that we actually take much more data for the training, and that's a simpler approach, because we don't basically, this validation building blocks can be similar to whatever else in other building blocks, because we don't restrict this time on the similarity at all. We just split on the identical building blocks. So what else here is I increased the amount of building blocks at each position by 3 times. Basically, this table will summarize it. And there are 5 folds. Also, I took all, like basically my positive subset is every molecules that has at least one positive activity to one of the targets. So that is a positive subpart of the data set. And the negative subpart is where a molecule always has a negative, and like basically it doesn't have activity to any of the targets. So that's all negative part. And I somehow mix the two so that I have the same distribution of the positive to negative ratio, the same basically ratio, positives to negatives, as in the validation sets, as I have in the original training data set. I mean, in the original data set overall, because it is important to model, to track the metrics in the same in balanced validation, because otherwise we might get too optimistic metrics and we cannot compare the models in this way, because the imbalance is huge. As I said before, the overall imbalance is basically 1.5% of positives over all molecules. So we try to model that. We try to be as close to that value as possible. So yeah, basically these are 5 folds. Quickly summarizing what we have. If we take 51 building block at position 1, we take 102 building blocks at the positions BB2 and BB3, and there are some tricks you can follow. Other discussions why we do this kind of intersection of building blocks and substructure. Subtraction of the two sets. That's kind of difficult to explain, but yeah, these are just the details. But overall, we try to roll and create different folds every time. Of course, we can do many more other combinations, but I just created 5 of them, and that was enough for me to model. So, and the training set here typically includes around 1.7 million molecules, and the training positive rate, basically positive to negative ratio here is 50%, because I can control that, and that's just for the training. That's good for the models. I don't need to apply any weights on my loss functions or whatever. Easy to actually experiment with, and for validation, as I said, I pick some molecules from any positive subset, and I also sample as much negatives as I can to be close to the ratio of 1.5 imbalance. So from the all negative sets. And yeah, these benchmarks, this splits also like work very well in terms of comparing models. And here I put the counter example where MPNN actually performed much worse than Malformer. That's what that's where I took the decision to not go with MPNN, like basically MPNN is a graph neural network, to not go with graph neural networks in my submissions, my selected submissions. Because on the high BB split, as you can remember, there were too many fluctuations for this MPNN model. And on the BB split, we can see that it actually underperforms. But here we can track it. We can better understand the performance of both models in comparison. So yeah, and overall, the private leaderboard score would be higher for the transformer models, and it's worse. Oh wait, here should be GNN++. Yeah, I believe it's the same amount of parameters. So basically, I tried to apply graph neural networks and transformers as my main hypothesis, and transformers performed better than graph neural networks. It's not just based on a few examples that I showed. It's based on like about 200 experiments I ran in total for this competition. So, and yeah, indeed, these models perform better. But to train Moldformer, it actually, well, I like training transformers because they are much simpler. It's much easier to just have a string of smiles and train on them rather than having to represent atom and bond features for the graph neural networks. So that's also kind of a useful like takeaway that we might switch and we might just use transformer models for scaling to billion size molecular libraries because it's easier, it's faster, it's more pure. You can throw it on let's say on A100 GPUs or TPUs and process, you know, with big batches to have some unsupervised pre-training and then fine tune. So that's where I see the direction for the later research. But anyway, if you want to try other models, I highlighted all of them that what I tried, and maybe some of you might find that actually graph neural networks will perform better than transformers, and which I actually, well, I expect that can be possible, like because there are so many combinations to do that. First of all, there are like a lot of different graph neural network architectures present. I just picked state-of-the-art models, which are GPS++ and MPNN++. There is a library Graphium available in open source that has all of those models implemented very well. So, and the combinations also might be in the features. And I took like this set of features, like atomic number, group in the periodic table, total valence, degree of each atom, basically how many atoms it is connected to, Fulmer charge, radical electron hybridization, chirality, number of hydrogen atoms, because we don't basically for each atom, I put a value of how many hydrogen atoms does it have as adjacent atoms, because we don't. It's better to not put hydrogens explicitly because it will enlarge your molecule so much it will slow down your training. But thinking about it as a feature, we can just you know, make it 3 times faster. Also, whether a certain atom is dedicated to aromatic ring and electronegativity. Also for bonds, we can encode bond type, whether it's stereo or not, and whether it's in ring or not. So we can try to play with different features, maybe remove some of them, and the model will not overfit that much. But it's kind of a lot of experience needed to be done to understand that. So maybe you will be able to do that. Anyway, graph neural networks are people try to scale them to billion-size libraries, and that's where in the paper on scaling of the models is written about. So this is again from the Valence Labs researchers. Dominic Biani is the leading researcher here. I met him in Montreal on the Machine Learning for Drug Discovery summer school, if you have heard about that one. This was this year. It took about 10 days, and it was very nice to see everybody there. So such a huge community where we were talking about many methods on in the field, specifically how machine learning can be applied in drug discovery. So this group of people, they try to scale GNNs to billion size parameter models and billion size molecular libraries, and they try to check whether they took 3 different graph neural network architectures. MPNN++ as the best, GPS++ as one of the state of the arts, and a transformer model, but not like the one that we use in our solution. And this is not smiles based. This is again the graph representational model, and the transformer is put on the nodes. So it's as they say, the architecture is identical to GPS++, but without the MPNN++ module. So it's just the pure positional structure encodings. So if we try to scale those different architectures, let's look at the let's say, let me check which plot is the best. Yes, let's maybe pick a molecular fracture. Basically, we increase our data set from 12.5% to 100%, and see how the models perform for different data sets. And we can quickly see that the blue curve is actually above all of the others. This is a loss function, so it's better in all of the cases mostly. And this is MPNN++. So this is the model without global self-attention on the nodes. So GPS++ is similar, contains the same building blocks, but it puts a global self-attention. And global self-attention might create a problem of, it might be a problem of, I would say, over squashing, if they call it. So you can find it in literature. There are 2 problems with graph neural networks: over squashing and over smoothing. And the global attention might involve over squashing problem. So, but and the transformer model is similar to this GPS++, because they have similar nature of the global self-attention. But that's hard to compare with smiles-based models, because that has nothing to do, and there's no comparison with those models. So maybe in our work we can continue doing that. So in our company, we try to train smiles-based transformers, and we have already achieved very good results with them. And I believe that might be a good direction. So I also tried other things. Let's say I created the ad hoc architecture called sim attention that is built on the similarity features between the molecule of the test set and the top 50 closest molecules in the training set. That's kind of ad hoc model. And also I trained the XGBoost models, as many people do, on RDKit features and ECFP features. So maybe I'm not that good in traditional machine learning, but I did not get any even close results to what I was getting with deep learning models. So, and I also try to train an MLP model on this RDKit, ECFP, and similarity features. Also didn't perform better. And a meta model, which might be basically an ensemble of all of the features plus the best performing malformer. Indeed, it slightly improves the quality on my benchmark, but I did not pick this model for the submission because it didn't look too obvious for me that this model outperforms. And actually, it introduced more complexity. It might even overfit slightly, and I will actually, the benchmark might not capture this this overfit. So I didn't select it for the submission. what I selected is what I actually showed on the final solution. That's pure smiles-based transformer models, and they perform the best. But there was other submission made by me 10 days before the competition ended. It actually performed on the score of top 3 position. If we just ignore if everybody would select the best, that score was also performed by a pure Roberta model trained on balanced set of positives and negatives. It was just about, if I remember, 3 million data points. And that model performed actually even better. So, but anyway, the main point here is that whatever I was selecting based on my benchmarks, and I'm happy that I chose the right ones, because the shuffle was really huge. If you can look at the leaderboard, you can see how many people got into the gold medal from out of nowhere, including myself. But this is what I was trying to do all the time. So some people got to these positions by luck, but I believe so I kind of deserved that position because I was working on the benchmark side a lot. And actually, I spent maybe 85% of my time building benchmarks, not models. So yeah, I think I want to finish at this point. If you have any questions, let's discuss.
Thank you very much. The great talk and so great results. Again, congratulations. So colleagues, please don't hesitate to ask questions or comments or suggestions, whatever. You can write in the chat, or you can unmute yourself and ask just by voice. Up to you, what is convenient for you. So
maybe I ask.
Yeah.
Yeah, sure.
Did you try to take one method of split, the best one maybe, and try to make different validation folds and compare scores, how they different between each other? I mean, same split and different validation folds.
Same split and okay, and basically, and when you want to check how the metrics look like,
what was the
right?
variability like diversity between
folds and
scores
between the folds, and how I believe you expect to see the metrics on leaderboard, right? You want to compare with leaderboard metrics. So how?
First of all, I wanted to know just diversity of local scores. And
I see.
how well they correlate to the leaderboard. Yeah.
Yes, that's a good question. Okay, actually, I have the results. It will be hard for me to find them right now in my DVC Live logger, but I can say that indeed there is a huge deviation between the folds. I actually did not try to like kind of improve on that. I don't even have any ideas how to solve this problem. But indeed, on different folds, there are very different results on the validations that I get. And my solution was from the beginning that, well, before building this 5 fold splits, I decided I will create an ensemble anyway. So whatever will be there, whatever spikes will be, whatever difference will be between the folds, I will create an ensemble, and that's it. So yeah, that's what I found.
And so you calculate the average, right?
Yes.
Average score.
Yes. So I picked the average. So I didn't train any meta model for the final solution. So it just includes the average of 5 folds for the BB splits and the average of the predictions for BB and high BB splits for the non-shared part. And for the shared part, this is just two models, Robert and Malformer, being overfit on all data, and also like taking average of the predictions.
And how did this average score correlate with leaderboard scores?
You mean, okay, you mean from which point, here or here or here?
For non-shared part.
For non-shared part, let's say I think the best what I can describe that would be through the same maybe post here, because yeah, basically I think because this is already an average, as you can see. For the high BB split, I took one best model. Oh wait, it's not like full average. All right, so okay, I don't have the average of BB and high BB models, but I have them separately. So this high BB and this one is BB average across 5 folds. So, and you can see that indeed high BB performs even better on the non-shared part over a building block model split. But we don't know if it's because of the average of the ensemble. Maybe some models perform so bad and it reduces the score. So there's no like clear picture. And I don't even know how to model that at this point, because data sets will be very different. The training data set are very different. So it's hard to actually compare side by side.
Yes, and also you did say, and we also noticed that for different folds, for non-shared folds, we had very, very different scores, and we could not compare at all.
Yes,
anything.
Yeah. Look, from my experience, from our company's experience that we got so far, is that it's very often that you will see, if you, let's say, retrospectively compare your predictions with, let's say, high throughput screening, like this one, you may get things close to random even, and that is specifically happens for the novel molecules in the non-shared, basically for the novel compounds. And well, it's actually a different idea here that I wanted to put. Maybe it's too complicated to discuss, but the thing is that sometimes it's easier to generate new molecules that models thinks are high binders, like that have high scores, rather than trying to retrospectively analyze and and compare the predictions with the ground truth values that you had in the past within your screening campaign. That's actually a not obvious thing, but look, let's say Leash Bio tested 100 million compounds, and if we retrospectively, we are getting like not so high quality metrics, I would say. They're not like crazily good, and moreover, we don't know what if we, because for the after the DNA encoded libraries, there is a follow-up step typically, which is a proper confirmatory bioassay test, and for that you have to pick just a few compounds, maybe 100, because it's an expensive test. And how would you pick this 100 out of 100 million? Well, you typically just will take the best scoring compounds, right? Maybe apply some clustering, but anyway, you will be taking the high scoring compounds. And what we found is that we may not need to try to compare top 100 best performing with the ground truth, because the scores will be so low there. But instead, we screen another library, which might be very big in terms of size. It might be, like n 40 billion, for example, what we do. So 40 billion molecules, we can screen them with this model and cherry pick the new ones, the new we will cherry pick the new molecules from the top, and we will hope for the better enrichment. And that happens. I mean, that indeed looks like that indeed works from what we found so far. That it is easier to take new compounds rather than retrospectively analyze your results.
Yeah, that's interesting. Actually, about new library, did you check scores for non-triazine only?
No, I didn't, because yeah, I didn't, because I was not modeling that part. I decided to skip the problem with the course because I didn't know how to tackle it, except of the fact that I decided not to work on building block level. I decided to work on the molecule level. So I think that's one of the assumptions that helped me, but nothing else, like specifically for course. I didn't do nothing else specifically.
Yeah, in this competition, this was not solved at all because scores are random, just random numbers for most of the participants.
Where did you find that? Did anybody publish such a post?
Yes, I asked in the discussions, and a few people answered. All have about 0.015, like so.
On the non-triazine core, right?
Yeah,
interesting.
only this part yet, and all have the same low score.
Oh, interesting. I think what also helped in my case, but okay, yeah, it might help, is the standardization. So I applied some SMILE standardization. Basically, I applied Kaggle structure pipeline for the molecules. It removes some salts. It removes some typical fragments that might be, not affecting binding affinity, and it also, of course, canonicalizes the SMILE string. And before application of the Kaggle structure pipeline, I also removed the dy point, whereas the linker of the DNA tag is connected. So I did that for I basically replaced dy point with methyl, what other some other participant did in the competition. So yeah, that's it. Any other questions?
Yeah.
I can tell it's very interesting, all your approaches, especially with different splits, because we had like twice bigger public scores for not shared part. If you show your table again.
Oh.
Yeah, this
one,
one.
right?
Yeah, this one. Like we have for our final model, like 0.2 for public non-share, twice more than yours.
No, really? For public
third.
0.2? Oh, you
yeah,
have
this, yeah,
0.2
yeah,
here.
this one.
Oh, interesting.
Yeah, 1.2. Yeah, and we have 0.6 on the left, private. Yeah, here. Lower. Private.
Yeah, I see. So
yeah, that's here you win, because we also have much higher shared scores.
Yeah. Interesting to know. Yeah, I believe that's what I was focusing all the time, like to not try to overfit on the public interval, because I was kind of familiar with this problem, because we are in the company trying to solve similar things. We want to adapt to a general model. to a more general solution. And yeah, I kind of knew that similarity affects so much, and we should be very careful about that.
Yeah, despite all tricky stuff with small samples and different scores, even locally and with faults, even average scores, anything. Anyway, your approach work. I see that. And also, I wanted to say, maybe will you share this code?
Code. I didn't go to that. I may share some, but yeah, I think the useful part. Okay, yeah, I may share something. I may share at least the notebooks for the data splits, right? That's the clear notebook I have. You will easily understand it. Yeah, and also maybe I will share some models, but I believe you can quickly find those models publicly available and just apply. Because
yeah, I want to share splits mostly. And
splits, okay, yeah,
yeah.
splits. I will share the splits. Yeah, of course. I will share it sometime later.
Good. Thank you very much.
Thank you for listening.
Okay, colleagues. So thank you very much, Vlad, again, and thank you everybody for listening. So I think we can finish for today. So Vlad is in our chat, and I think if any questions, we can ask Vlad in the chat. So the video record of this webinar will be soon on YouTube, and again, the link will be in our chat. So thanks again, and congratulations, and we wish you great successes in the future. More gold models, oh, sorry. Gold medals.
And you too. Everybody, yeah, everybody. Let's solve this generalizability problem on small molecules at least, and maybe next move to other modalities.
Yes, it's very important. Okay, so thank you very much.
If you need any information from me anytime, message me in Telegram or in the group, because I also in this cyberlogger group. So yeah, let's discuss things there. Always happy to collaborate.
Okay, thank you very much. Thank you very much. Bye-bye.